This method addresses a common problem within the field and provides a complete workflow for rapid isolation and characterization of single extracellular vesicles with specific markers of interest. Nanoparticle-tracking analysis is a semi-automated method that utilizes the properties of light scattering and Brownian motion to analyze the size distribution of extracellular vesicles. Demonstrating the procedure will be Vera Schmidt, a PhD student from our laboratory.
To isolate the extracellular vesicles, after collecting 2 milliliter samples of human whole blood into serum separating tubes, allow the samples to coagulate for 15 minutes at room temperature before separating the cells from the serum by centrifugation. Transfer 1 milliliter of serum from each sample into individual 1.5 milliliter reaction tubes for centrifugation to remove the platelets. And transfer 100 microliters of the platelet-poor plasma to new 1.5 milliliter reaction tubes. Next, add 25 microliters of exosome precipitation solution to the plasma and vortex the samples thoroughly. After a 30-minute incubation on ice, collect the extracellular vesicles by centrifugation. The pellet will appear as a beige or white color. Aspirate the supernatant and centrifuge the sample again. Then remove all traces of fluid and resuspend the pellet in 100 microliters of PBS. To stain the cell